Hi everyone, thank you for joining us today. My name is Janelle and I'm here with Elad, and we are going to talk about acquisition and analysis of reproducible high-complexity cytometry data using the Cytek Aurora and the Astrolabe platform. To kick things off, I'm going to talk about the Cytek Aurora platform and tell you a little bit about our approach to panel design, how to select reference controls, and how to evaluate the performance of your panel on the Aurora. Then I'll hand the presentation over to Elad, and he'll show you some of the analysis tools available to you in the Astrolabe platform. For the 5-laser Aurora, it comes with a 355 nanometer laser, 488, 405, 561, and 640. All of these lasers are spatially separated as they pass through the flow cell. From there, any emitted light from your sample is collected by the fiber optic cables and passed down to the APD detector modules. These modules are highly sensitive and have low noise, and there is one array dedicated to each laser line. Additionally, there are 2 side scatter detectors on board, one off of the violet laser and one off of the blue. To the right of each APD module, we've drawn a little schematic showing the filter sets that are within each module. Each module can have up to 16 detectors inside, and each detector has a different filter in front of it. Together, they capture light across a range of wavelengths, starting after the wavelength of the laser and stopping around 830 nanometers. Now let's look more closely at these detector arrays and think about what happens when a particle passes through the system. If the particle passing through is bound to a dye like BV750, which is excited by every laser on the system, you'll see signal in every single detector array, as shown. Now if we look at this top spectrum plot, the x-axis here is showing each detector in the array, beginning with UV1 and ending with UV16. The y-axis is showing median fluorescence intensity. The Aurora obtains a measurement from every single detector across all of the modules on the system. Then that information is stitched together to create what we call a full spectrum signature. To understand this plot, let's take a look at one of the channels, say V14, where this dye is maximally excited. In this channel, imagine that you are looking down at the top of a histogram plot. The red color represents the peak of the histogram, where the highest density of events are located. Then, as the histogram tails off, the decrease in event density is represented by this yellow to green to blue coloring. We have this type of information for every channel on the system, and for each dye that you run on the system, you will generate one of these signatures. The software then uses this information to unmix the data using an ordinary least squares algorithm to figure out what is going on in your multicolor tube. These signatures matter for us because it allows us to use highly overlapping dyes together, so long as they have some difference in their signatures on our system. As an example, let's look at APC and Alexa Fluor 647. If you were to run these on a conventional system, where you are only collecting the light for these with a single detector and a single bandpass filter, you might find that these dyes look the same on that system, and you cannot tease them apart. However, when you are collecting that light across as many as 64 channels for our 5-laser system, you might see some differences between these 2 dyes. For APC here on the top, you can see that it is excited by every laser on the system, as there is a lot of signal showing up in each detector module. But when you look at Alexa Fluor 647 on the bottom, there is very little signal in the violet and blue arrays. All you can see here is a little autofluorescence signal from the sample itself. When you look at the yellow, green, and red arrays, there's a lot of signal here. If you compare the yellow-green information between these 2 dyes, you can see that the pattern looks different between the 2 dyes. And the same story for the patterns that you see in the red arrays. So, since these 2 full spectrum signatures are unique, we should be able to use them together on the Aurora. What we did next is we made a small panel to demonstrate using these dyes together, and chose markers that are co-expressed on the same cell type. So what we have here is a simple 3-color panel, with CD3 FITSI, CD8 on APC, and CD56 on Alexa Fluor 647. The first plot is gated off of lymphocytes, and from there we gated on CD3 positive T cells and display those events on a CD56 versus CD8 plot to find the NK T cells. Then over to the left here in the green and gold plot, we are looking at that same CD56 versus CD8 gated from the non-T cells to find the NK cells in the sample. Even when using these two highly overlapping dyes together in this panel, we still see excellent resolution of our positive and negative events, and no resolution is sacrificed in visualizing the different levels of CD8 expression on the NK cells. Another advantage to using this approach of the full spectrum cytometer is the ability to characterize and work with autofluorescent samples. Here are a few examples of full spectrum signatures for different samples provided by some of our collaborators. We've got splenocytes here on the top, lung cells in the middle, and yeast cells at the bottom. When you compare these different samples, it's easy to see that some samples, like these splenocytes, have little autofluorescence, and other samples, like the yeast cells at the bottom, have very high levels of autofluorescence, which can make it difficult to see markers labeled with dyes that emit in these regions, as the autofluorescent signal tends to mask those signals. But in our full spectrum world, we can use this ability to measure the autofluorescent signal to our advantage. For example, the sample of M. cherry expressing HeLa cells shown here was provided by Malt Paulsen from the flow cytometry facility at EMBL. On the left, we are showing the unstained sample, and you can see it has quite a bit of autofluorescence here in the violet and in the blue arrays. Underneath it is the M. cherry single stain control, which is very similar compared to the unstained in the violet, the blue, and the red arrays. But when we compare the yellow-green patterns, we can see there is a slight difference between the M. cherry and the unstained signatures. So, if we record these tubes and unmix them, what will we see? In the first plot here, we have our unstained tube showing side scatter versus M. cherry fluorescence, and the second plot shows the sample tube. But we can't really tell where the M. cherry positive events are, as the data looks a lot like the unstained tube. So what we can do is we can re-unmix the data, but this time extract out the autofluorescence. Now we can clearly see the positive M. cherry events and distinguish them from the cells that are not expressing M. cherry. Now that you understand how the system works, we can now talk about how to design a multicolor panel for the Aurora. In order for you to be successful at this, the first thing you must do is gather as much information as possible around the biology of the assay. The more you know about your sample, the more informed decisions you can make later on when you're trying to pair markers with fluorochromes and create a high-quality, well-resolved multicolor panel. When you're doing this, there are two things you want to look for. One is you want to figure out how to classify each antigen, which we will cover in the next slide in more detail. Second, you want to understand which antigens are co-expressed on each of the cell types you want to look at. This is going to be very valuable information when deciding which dye to pair with each antigen, so you can clearly resolve all of the co-expressed markers in your panel. When thinking about antigen classification, we highly recommend that you read this Roederer paper. It talks about 3 different levels of classification: primary, secondary, and tertiary. The primary level are the antigens with the highest levels of expression. For the human PBMC panel we are looking at today, the T cells we are looking at have primary antigens of CD3, CD4, and CD8. The secondary level of antigens have slightly lower levels of expression, and they often show up as a smear of staining in the dot plots, as there's a gradient of expression levels within the cell type. Some examples here include CD45RA and CCR7. Also, for some cell types, like our NKT cells in today's panel, markers like CD8 can be used for subsetting these cells, and it has a lower level of expression on this particular cell type compared to the T cells we discussed earlier. Last but not least, the tertiary level are the antigens with low levels of expression or a very low frequency of occurrence, and hence they can be the most difficult to detect. So we want to make very smart choices when deciding which fluorochromes to pair with each of our tertiary antigens to ensure we can clearly resolve them in our multicolor tube. Once you've gathered all of that information, you need to summarize it in a way that allows you to easily see all of the different primary, secondary, and tertiary antigens in your panel, and helps you to understand and see all of the markers that are co-expressed on a single cell type. For example, if we focus on the cytotoxic T cells in the blue rectangle here, we can easily see all of the markers that are co-expressed on that cell type. Now that we have all of our information summarized, it's time to start deciding which fluorochromes you want to assign to each of the antigens. To do this successfully, there are a few things you need to consider. First, you need to choose fluorochromes that all have unique spectral signatures. Second, you need to understand the brightness of each dye so you can pair it wisely with each antigen. And third, you need to understand how these dyes interact with each other in a multicolor setting, as these interactions can introduce different levels of spread into your data. We'll show you a couple of different tools that can help you understand spread and help inform your decisions in pairing each antigen to a fluorochrome. Starting with our first point, let's take a look at some signatures to see which dyes could be used together in our panel. The example shown here is from one of our fluorochrome selection guides and shows dyes excited primarily by the blue laser. Each plot represents a fluorochrome with a unique signature, so you can use dyes like BB515 with Alexafluor 488 shown beneath it. Dyes that have identical signatures share a single plot with the names of the dyes written together on that plot. Looking again at the BB515 signature, we can see that BB515 and Bio515 have identical signatures, so we cannot use those two dyes together in a panel. Once you've chosen dyes that are unique and can be used together in a panel, you need to consider the brightness of each of these dyes when assigning them to your antigens. What we are showing here is a ranked order of dye brightness based on the dye's stain index. The dimmest dyes are furthest to the left, and the brightest dyes are to the right. This type of chart will vary a little by system configuration. This particular chart was generated from a system with a yellow-green laser, and you can see that PE is one of the brightest dyes on the system, all the way here to the right. When you start thinking about how you want to pair each of your dyes with your antigens, you need to refer back to how you classified each antigen as primary, secondary, or tertiary. When pairing the tertiary antigens, you want to use the brightest dyes you have for these, so you have the highest chance of detecting them in your panel. For the secondary and primary antigens, you compare the medium bright and dim dyes with these, as those antigens will be easier to resolve given their higher levels of expression compared to the tertiary antigens. Don't forget, there is still one more thing you need to consider. We need to think about the spread that these dyes could have into each other in the multicolor setting. There are a few different ways you can think about spread. One way to glean a lot of information about spread is to look at the full spectrum signature of the dyes. What we are showing here is the full spectrum signature of PE-Sci5 up on the top and of APC on the bottom. We can see that both dyes are excited by the violet, the blue, and the red lasers. You can also see that APC is maximally excited by the red laser, as it's peaking around R1 and R2. PE-Sci5 is maximally excited by the blue laser, as its peak is appearing around B7 and B8. Both dyes, while they are not maximally excited by the violet laser, do have a good amount of signal showing up in the violet detectors around V11 and V12. Now, if we take these signatures and normalize them so they're now on the same scale of 0 to 100%, we can learn a lot about how these dyes will interact with each other. For example, if I look at this first rectangle around V11, V12, we can see that both dyes, APC shown in red and PE-Sci5 shown in blue, have some amount of signal in this area. What this means is that these two dyes will have some spread into dyes having maximum emission in that part of the spectrum, such as BV650 or BV711. Then, if we move to the middle rectangle, which is around B7 and B8, we can see that APC has very little emission here compared to PE-Sci5, which is dominating in this region. What this means is that APC will have little spread into PE-Sci5. Next, we go to the last rectangle, which is around R1 and R2, where APC is dominating. But we are still getting a lot of light here from PE-Sci5, and what this tells us is that PE-Sci5 will have a lot of spread into APC. How do we see this in the multicolor data? To demonstrate this, we prepared single-stain CD4 cells in APC and also in PE-Sci5, then mixed them together in the same tube and recorded them on the system. And here's what you see. When you look at the APC positive events, you see a nice tight cluster with only a little spread into the PE-Cy5 axis, which is what we were expecting based on what we were seeing in the normalized spectrum in the B7, B8, B9 region. Then if we look at the PE-Cy5 positive events, we can see that there is a fair amount of spread into the APC axis, which is also what we were expecting based on our observations from the red rectangle. This example is only showing the interaction between one pair of dyes. How do you understand the interactions occurring between all of the dye pairs in your panel? Lucky for you, there are tools available to help with this. One such tool is the spread matrix tool. The way to read this is you look at a fluorochrome in the row, and this fluorochrome will impact the dye in the column. If there's a deep pink or dark red color for that dye pair, this means the dye in that row will have a lot of spread into the dye in the column. If there is a white or light pink color for the dye pair, this means the dye in the row will have little spread into the dye in the column, which makes these dyes very easy to include in your multicolor panel without sacrificing resolution. Let's consider our PE-Cy5 and APC example again using this matrix. First, I'll find APC in the row and follow it all the way over to PE-Cy5 over here. And what I see is a medium pink color, meaning there will be some spread from APC into PE-Cy5, but not much. Next, I'll find PE-Cy5 in the row and follow it all the way over to APC, which is over here. I see a really dark red color, meaning PE-Cy5 will have a lot of spread into APC, which agrees with what we were seeing on the previous slide. In SpectraFlow version 2.2 coming out this month, there are a couple indices we've added to help you assess spread in your panel. First is the similarity index, which compares each dye pair in your panel and gives it a value between 0 and 1. Any values that are close to 0 means the two dyes are very distinct from each other. Let's look at a couple of examples. One here is FTSE and APC Cy7, whose similarity index in this panel is 0. So these two dyes are very unique and have little to no impact on each other. In contrast, if we look at BB700 and PerCP Cy5.5, we see a similarity index of 0.98. So these two dyes are very similar to each other and will likely have a lot of impact on each other in the panel. The other index we provide is the complexity index. This index is an overall metric for the entire panel and gives you a relative number that tells you how close all of the dyes in the panel are together. The smaller the number you are able to achieve, the better the combination and the less spread you will see in your data. Let's look at a few examples. You might be able to make a 10-color panel on a 3-laser system using mainly non-tandems and dyes with low amounts of spread into each other and get a complexity index of 2.3. Say you then add 10 more dyes to your panel to get to 20 colors, you might get a complexity index of 9.8. If I add another 6 colors, I could get a complexity index of 36.5. One way to use this tool is to compare panel iterations. Say you want to have a 20-color panel, you record it on the system, and you get a complexity index of 9.8. Then you decide to make a couple of changes in the dyes you are using, run the panel again, and get a new complexity index. If that index is less than 9.8, you've improved your panel. But if that index increased, you likely introduced more spread into your panel. Once you've made all of your fluorochrome assignments, it's time to put it all together and start evaluating your panel on the system. We recommend organizing your panel in a table like this one, where in each column you are organizing the reagents by the laser they are maximally excited by, and then ordering the dyes from lowest to highest maximum emission wavelengths. This is helpful for two reasons. One, you're able to look in the laser line and see which markers are closest to each other. Second, when you look across the columns within a specific emission wavelength, you can see how many dyes in your panel are emitting a lot of light at that wavelength. This is important to look at because there are many dyes that are excited by multiple lasers, so you can easily see these challenging areas and consider the effects on your panel and plan for them. Next, you need to make sure that you choose the right controls for your panel. To do this, we have 4 general guidelines you should follow. First, you need to make sure that the negative and the positive particles are clearly separated. Then, you need to make sure those particles have identical autofluorescence characteristics. You can't use a positive cell particle with a negative bead particle and expect this to work out on the system. Next, you need to make sure you record enough events for both particles. If you only record a few hundred events, it's going to be challenging to get an accurate estimate of the median fluorescence intensity for those particles. But if you record a few thousand, or even 10 or 20,000, you'll have a much better sample size to work with. Finally, you need to make sure that the fluorescence spectrum of the positive control is identical to the one in the multicolor sample. If it's not, you're going to encounter unmixing errors in your multicolored data that you'll have to manually correct. In SpectraFlow version 2.2, we have a new tool to help you QC your controls. Remember we introduced the similarity index a few slides back to compare the uniqueness of different dyes to each other. We can also use this index to compare the control you acquire today in your experiment to a gold standard or benchmark control and tell you how similar or different they are. If you come out with a similarity index close to 0, the software will flag that control as it doesn't match the benchmark control. For example, this top plot is showing FTSE signatures, where the red line is the benchmark control, which shows a lot of signal around B1, B2, and B3. The black line represents the control you acquired today in your experiment, which here looks very different from the FTSE benchmark control and shows a lot of signal in the red array, so it's likely that the wrong dye was acquired. The other two examples here are for PE and for APC. For both of these, you can see that the controls acquired in the experiment today align very nicely with the benchmark controls, and consequently, their similarity indices are very high at 0.99. Once you've recorded all of your controls and your sample, it's time to see how accurate the unmixing is. To do this, there are two steps you should complete. First, you should check the unmixing accuracy of all the single-stain cells. To do this, go to one of your controls, like this Alexa Fluor 488, and plot it against every dye present in the panel. What you want to see is this nice, properly compensated appearance in the data in every single plot. Sometimes you may find strange patterns, such as the positive events are curving upwards or curving downwards if they are not compensated correctly. We see this happening sometimes when beads are used as reference controls, and the spectrum of the dye when the antibody is bound to the bead is not identical to the spectrum of the dye when the antibody is bound to the cell. Remember, your single-stain cells are your gold standard and will guide you on the decision for which particle is the best choice for each of your controls. The second step you need to complete is to check the unmixing accuracy in your multicolor tube. In this example, we're showing per-CP Psi 5.5 on the x-axis against all the other colors in the panel. Something to look for here is the presence of aggregates in your data, which will appear as very bright events in the upper right portions of the plots. You could gate out the aggregates to clean up your multicolor data, and in the future, use this information to know which dyes you'll need to spin down to avoid seeing aggregates in future sample preparations, or even consider changing that reagent entirely if it keeps causing problems. There's one more step to complete to finish optimizing your panel, and that is to look at all of the single-stain cell controls and compare them to the multicolor tube to evaluate the performance of your multicolor panel. There's a lot you can learn by going through this exercise. For starters, you can see if your antibody titer is optimal. If you notice a shift in the MFI for the positive events in the multicolor tube compared to the single-stain control, your titer may not be quite right. You can also see if your reference controls are working out well and unmixing the data nicely, and assess the quality of your panel design. If we look at the cartoon here on the left, the top plot is showing marker A on Fluorochrome X for the single-stain control tube, where we see a nicely clustered positive and a tightly clustered negative, with a good amount of separation between the two. Then we look at that same marker for the same fluor in the multicolor tube. We notice there are some changes. The biggest one is that the negatives have quite a bit of spread introduced into them once they are in this multicolor environment. We can still differentiate our positive events from our negative events, so we can likely live with this amount of spread in the negative. But these are the types of decisions you need to make in your panel as you try to optimize it. Taking the time to compare each marker in the single stain tube against the multicolor tube is the foundation for panel optimization. Now let's look at a couple of real examples. This first one is showing CD56BV421, where the top histogram is from the single stain control and the bottom is from the multicolor tube. When we compare the two histograms, we can see that the negatives are pretty similar. The positives are showing up in similar locations, and our ability to resolve the different levels of CD56 expression from the negative events are very similar between the tubes. If we look at another example, say CD28BV785, here's how the single stain control looks, and here's how the multicolor tube looks. For this one, we get a little more spreading of the negative, and we are starting to lose that nice clean separation between the positive and the negative cells compared to what we are seeing in the single stain control. What we need to decide is if this is something we can live with. Can we still easily gate on the events that we want to see? Or is this something that we need to improve by making changes to our panel? Once you've completed this exercise for all of your dyes and you are happy with the resolution results between your single stain controls and your multicolor tube, you are ready to start analyzing your data. One way to go about this is to use our SpectraFlow software to make pseudocolor or dot plots like these and gate down through your data to visualize all of your subsets. But there are many other analytical tools out there, one of which is the Astrolabe Flow Cytometry Platform that Elad will now take us through.
Thank you, Janelle. I've been working with Cytek Aurora data from different organizations for over a year now, and I'm repeatedly impressed by the high quality of the data. It's really fun to work with. So hey everyone, I'm Elad, the CEO and co-founder of Astrolabe Diagnostics, and following Janelle's discussion of panel design and quality control, I would like to discuss some of the analytics for the Aurora data, specifically using the Astrolabe Cytometry Platform. And just a quick note, if you have any questions, please use the Q&A button below. We're going to address all the questions, or at least as many questions as we can, at the end. So one of the common stories that I encounter is that researchers are really excited about a new technology, such as the Cytek Aurora, and they acquire the data and everything looks good, but then when it comes to the analytics, they get a bit stuck. And there's different algorithms out there to help you. There's Visney and Flowsum and heat maps and the R programming language. But despite this rich ecosystem, the learning curve is still pretty steep. And once you know how to use these tools, applying them to your data can be quite time consuming. In my mind, what researchers want is some way to take their single cell data and process it and reach meaningful biological insights, something you can use for your next grant application, your next paper, and ideally to impact patients' health. And one way to do it is manually, either through traditional gating or using these algorithms that help you tidy up the data, help you structure the data, but there is still a significant manual component to processing it further. What Astrolabe offers is a way to deal with the mechanics of single cell analysis in just a couple of hours. The Astrolabe platform is an automated consulting service where you upload your data, you explain your experiment design, the same thing you would do with a bioinformatician. And after you do that, the platform kicks in, takes care of the analysis, and you can navigate around the results through a user-friendly cloud-based interface where everything is point and click and the insights are right at your fingertips. Astrolabe can deal with human and mouse data. It can deal with many different tissues, including blood, lung, brain, and others. And so far, we've analyzed healthy data from healthy donors, cancer, autoimmune disease, vaccination studies, and many others. One of the critical parts to me is that Astrolabe is not a faceless software company. We're very keen on what we call research or partnership, where if you have any questions about the analysis, what does this cluster mean? can you tell me more about the statistical analysis? Is this real signal or just noise? We're here for you. So you can always email us with scientific questions, with challenges that require intellectual engagement. And we're going to be there for you. So the platform has many different features. We can clean the data for debris and doublets. The platform does quality control, cell subset labeling, unsupervised clustering, and all the follow-up analyses, including dimensionality reduction, differential abundance, and differential expression analysis. All of these are available through the online interface, but also through various export formats, including FCS files, spreadsheets, PDFs, and R files. So it is quite a lot. And for the purpose of this webinar, I'm going to follow up on what Janelle started with and talk about quality control and some of the initial steps of the analysis that you might want to look into. So I'm going to switch to the Astrolabe platform and show you how to analyze your data. So I'm going to start a new experiment, and I'm going to call it Astrolabe demo, Cytek Aurora data. And I'm going to click here to upload some Aurora files. And this is a 31 color study that we conducted in collaboration with Cytek. So the files are uploading, and once they are done, I am going to click the Find Features that appears at the top right. At this point, Astrolabe imports the files and makes sure that everything is right, that the panel is there, that no files are corrupted. It does take a couple of minutes, so I'm going to skip that. The next step is to set up your experiment. And the first thing you do here is you instruct Astrolabe on which channels you would like to use. And I've picked all of the different surface markers channels, and if I wanted to add or remove something, I could do it here. Furthermore, if I wanted to rename any of the channels, let's say if we had a typo, then this is where we could fix it. Additionally, at this point, we could specify any features for the statistical analysis. So this study is just 5 healthy donors, so there isn't really much of a research question here. However, if we had, let's say, controls versus treatments, so we could add a feature called control, and it's going to have control versus treatment. And we could classify the samples accordingly. And then let's say we had 3 different doses of the treatment, then we could add a dose, and I'm just making stuff up now. So we could add a dose and set it up here and tell the platform these are the different doses and these are controls. So they don't have a dose at all. So you would set up your experiment design, you would click next, and Astrolabe is gonna analyze the data, clean it up, find the subsets, and so on. This process usually takes somewhere between 30 minutes and 2 hours. So once again, I'm actually going to skip it with an experiment I prepared in advance. So this is the results of the experiment, and there's different tabs here and different places you can go. I'm going to start by clicking on quality control. Astrolabe passes your data through various quality control metrics. For example, the platform identifies debris using forward scatter and side scatter. And you can see the percent of debris in each sample. These are all PBMCs, and this debris count is reasonable. But if anything would have gone wrong with any of the samples, you would immediately see it here. Additionally, Astrolabe is looking for missing subsets. So for example, if your panel has CD3 in it, but Astrolabe can't find any T cells, it's going to highlight it here. That might be a staining issue, or maybe the sample really doesn't have T cells. It's up to you to decide. Speaking of staining quality, Astrolabe implements the average overlap frequency, which is a metric that was developed during my time in Mount Sinai. The AOF is a value between 0 and 1. The lower it is, the better the staining quality, and it was explicitly designed for the assessment of markers with bimodal distributions, that is, markers that have positive and negative signals. And looking at this heat map, you can see that almost all the values are low, around 0, showing that the staining quality on all of these surface markers is pretty good. You will also notice that ICOS and PD1 have more varied staining quality. And the reason is that not that there's a staining issue, we actually check these manually to verify that they look good, but because they're not bimodal. PD1 and ICOS tend to have a long tail, which is not suited for this specific metric. So you can easily assess the quality control of your data. And let's talk about the analysis itself for a few minutes. So what I'm showing you here is a tab called the Cell Subset Navigator. And this is a composite view of the entire experiment. Everything we have here is using data from all of the FCS files, whether you have 5 or 50 or 500. And the introductory view to the Cell Subset Navigator is a visualization called the MDS map. In the MDS map, each one of these bubbles is one of the cell subsets in the experiment, and their position on the map corresponds to the distances between them in the high dimensional space. This technique is known as dimensionality reduction. It's very similar to FlowSum or t-SNE or SPADE. And just to demonstrate, you can see that similar subsets like these T cells are next to each other, while these similar subsets like the T cells, the myeloids, and the B cells are far away. So from the get-go, Astrolabe allows you to use the language of immunology when dealing with the data. There's no anonymous clusters to annotate. There's no biaxial plots to label, such as t-SNE and UMAP. Astrolabe can identify the B cells and different T cells and so on its own. And the way it does this is by clustering the data and then labeling the clusters using a series of rules that depend on your panel. So if I go into the Quality Control tab and click on Labeling Strategies, Astrolabe is going to show me how each of the subsets is found. So, for example, looking at the naive CD8 T cells, Astrolabe first removes the myeloids with side scatter and CD33, then finds the B cells, then the CD8 T cells, and so on. Finds the T cells, and then the CD8 T cells, and so on. So the rules are available here. You can always go and make sure that they made sense for your experiment. And I think this is a good point to mention the importance of side scatter in this high dimensional data. It is a very powerful tool to separate the myeloid cells from the others. So it's one of the first steps that Astrolabe is using for the analysis. So going back to the Cell Subset Navigator, I would like to mention that this view is fully interactive. So for example, we can color the different bubbles based on the markers. And here's CD3, so we can see all the CD3 positive bubbles, all the T cells up here. And if we want to look at CD19, then here's all the B cells. We can also click on the cells to learn more about them. So I'm going to click on the naive B cells, and I'm going to click on the central memory CD4 T cells. And when I do, I can scroll down, and Astrolabe provides us with additional visualizations, additional information. So for example, looking at the naive B cells, we can see that they're between 2.5 and 5% across the 5 patients. So there is some heterogeneity in their frequency. And we also have a heat map that shows us the signature of these cells. So we can see that they're CD19 positive and CD20 positive. And for example, CD3 negative and CD56 negative. So all the trends that we would expect. Going further down, we can now look at the central memory CD4 T cells, and they're somewhere between 8 and 18% across the 5 donors. And looking at the heat map, again, there's trends we expect. There's CD3 positive and CD4 positive and CD27 positive. We can also see, for example, that they seem to be PD1 mid, at least in aggregate, and that there is some heterogeneity across the patients. For example, looking at CD127 and looking at CD25, their intensity varies between the different patients. Furthermore, again, this experiment doesn't have any research questions. These are just healthy donors. But if we had separated the patients into, let's say, control and treatment, then we would be able to color code these plots and we would be able to run statistical analyses to compare them. So looking a bit further at the heat map for the central memory CD4 T cells, I mentioned this heterogeneity for the different markers. And I think one of the exciting things about the Cytek Aurora is that you can include these additional markers that have biological significance, but are not as easily characterizable or as easy to utilize as, let's say, CD19 and CD56. And how can we incorporate them into the analysis? So to do that, Astrolabe offers an additional tool called cell subset levels. The bubbles that you see here, they all come from the assignment level, which corresponds to the to canonical subsets that are defined using the rules I've shown you a couple of minutes ago. The second level is called profiling, and when I click on it, you will notice that I have many more bubbles. And what happens here is that Astrolabe takes each one of the subsets and further stratifies it using unsupervised clustering. So we took the myeloids, and these are all still classical myeloids, but monocytes, but they've been broken down into different clusters. Same for the B cells, same for the central memory T cells. So we see that there's about 8 or 10 different clusters that originate from the central memory T cells. And scrolling down, as before, we can learn more about them. So for example, here's one of the clusters that Astrolabe found, and it seems to be much higher in this patient compared to the other three. We can look at the heat map to see what's the signature of this cluster, and also Astrolabe is using a heuristic to try and characterize them. So these markers, they're not the absolute truth on what these clusters are. It's just a heuristic that Astrolabe provides to make life easier for us, and again, to make communication easier with other researchers who might be working on this project. So for example, this cluster seems to be high for CD161 compared to the others, and high for CCR6 and high for CXCR5. Scrolling down, here is another cluster, which is overall more common than the first cluster, and seems to be lower in patient 5036, second most frequent in, second least frequent in 4395, and then about the same for the other patients. And scrolling down, looking at CXCR5 levels, these differ between this and the first cluster, and we can look at the heat map and see how everything else is about the same. Both the canonical markers like CD3 and CD4, and the profiling, the clustering markers like CCR6 and CD161, except for CXCR5, which is higher for the first cluster and lower for the second cluster. And as I mentioned before, if we did have any information about an experiment, let's say responders versus non-responders, we would be able to overlay it here and do any follow-up statistical analysis. So we can keep navigating around these different clusters. However, I would like to take a step back for a minute and look at a specific sample instead of the experiment as a whole. And to do that, Astrolabe offers a visualization called the Immunophenotyping Navigator. This is a sample-by-sample view, and it shows us information just about this sample. So we have all the quality control metrics from before, and we have a heat map that shows us the signature of the various cell subsets that Astrolabe found just in this sample. So we can see different B cells and T cells and myeloids and NK cells. And we can also use this visualization to create additional figures and dissect the data a bit more deeply. For example, if I choose two of the T cell markers and the naive B cells, two of the T cell subsets and the naive B cells, and then click on CD3, I'm going to have a density plot showing me the level of this marker across the three cell subsets. if I then click on CD19, then within a few seconds, Astrolabe is going to provide us with a biaxial plot where we can clearly see the B cells at the bottom right and the two T cell populations at the top left in purple and green. We can do the same thing for the profiling level. So now we're seeing all the different clusters that Astrolabe found. And beforehand, we saw that the central memory CD4 T cells were broken down based on the unsupervised clustering. We would like to inquire what does CXCR5 high and CXCR5 low means. So we can just pick the clusters and then click on CXCR5 to get the density plot and see what is high, what is low. And again, if we want the biaxial plot, we can just click on CCR6. And within a few seconds, we're going to have the biaxial plot showing us the separation across CXCR5 between these two clusters. So this has been a brief overview of Astrolabe's main capabilities. As I mentioned in the slides, we have a whole bunch of different features whose goals are to escort you across the entire analysis process, from cleaning the data through quality control, labeling, clustering, and follow-up statistics. And the emphasis is really on liberating the data and allowing you to get the most out of your Cytek Aurora experiment. So just a brief summary. One, I hope I managed to show you how easy and accessible everything is with Astrolabe. It's really about navigating through the data seamlessly and learning more about the biology and the immunology of your experiment, instead of having to deal with the raw data with the single cell data. The analysis itself is very fast. You put your FCS files and Astrolabe, wait somewhere between 30 minutes and 2 hours, and then you can come back and navigate through these high-level insights, focusing on the biology of your experiment. And finally, I mentioned the researcher partnership aspect, where if you have any questions, if you get stuck, you always have a place to turn to. You can always email us with questions, with advice, with ideas, and we're here to help. So just a reminder, if you have any questions, please use the Q&A button below. We're going to take a few seconds break to review the questions and then get back to you and answer as many as we can. So once again, thank you to Janelle, and thank you for Cytek for partnering in this webinar. And we're going to address some of the questions now. Janelle, can you hear me?
Yes, we can.
Okay, that's great. We didn't mention before, but in addition to Janelle, we're also joined by Gil Reining, the Director of Marketing at Cytek, and Maria J. Mess, the Direction of Applications there. And we have a few questions coming up. The first one for Astrolabe is a great presentation. How would we analyze data if it has abnormal immunoprofiles? And there's a few ways to do that, and they all come down to the profiling level that I've shown. So if we go back to the cell subset navigator, any cell that slips through the crack and doesn't end up in any of the canonical subsets is going to end up in these different groups called unassigned. And you can see this experiment has several of them, such as B cell unassigned and CD3 positive unassigned, and root unassigned, which corresponds to cells that didn't match any of the canonical markers. And these are part of the analysis as well. And if you have any unique subsets, more often than not, they end up there. And you can cluster them, and you can run the analysis on them. And in addition, if your system is completely abnormal, for example, if you're looking at AML, you can instruct Astrolabe to skip the assignment level entirely and just jump straight to the profiling. And in this case, everything will be clustered using unsupervised clustering, and you can continue the analysis from there. So the next question is for Cytek, and that is, what is the difference between compensation and unmixing?
Okay, so this is Maria. So great question, compensation and unmixing. So we can think about compensation as a simpler way of unmixing. When we do compensation in a conventional cytometer, we have a square matrix, meaning that you have n number of fluorochromes going into n number of detectors. So that's an n by n matrix. As Janelle explained at the beginning of the webinar, when we have a spectral analyzer, you often, very, very likely, will have a higher number of detectors compared to the number of fluorochromes that are in the assay. In the case of the data presented today, for example, we have a 33 color assay and we have an instrument that has 64 channels. So in that case, our matrix is not a square matrix, it's a rectangular matrix. So the mathematics to resolve that matrix are slightly different to what you use in compensation. The base, the foundation of the math is the same, but the algorithm that is used, that is in our case a linear regression, is slightly different to the algorithm used in classic compensation. Also, you can think about unmixing, where we are feeding these references that Janelle beautifully presented. We have the spectrum for each dye, and then in the multicolor sample, we have a mixture of all those spectrums. So what we're trying to do, what we do with the unmixing algorithm, is to understanding that mixture that is in the multicolor sample, which of the individual signatures fit. So mathematically, there is commonality, but there are also differences because of the kind of matrix that you are resolving. In the big scheme, the information that you have to resolve the matrix is different, in the sense that in the unmixing spectral analyzer, you have way more data that you are going to utilize to determine, in a complex mixture of colors, what is the contribution of every color. I hope this answered the question.
Yeah, and I actually encounter this question pretty often when talking about the Aurora with researchers, and I think that the best way to explain it is that there's no magic here, and as Maria pointed out, much of the concepts for unmixing have existed for decades through compensation. So it's a variant, it's different mathematics, but conceptually it's very similar, if I understand it correctly. So the next question is for Astrolabe, and that is for markers with dim expression, how is determination of positivity assigned for the different subsets? And the answer is that this follows the same concept of any other marker. And the algorithm underlying Astrolabe is a clustering algorithm called FlowSum. And there has been a benchmark published by Weber and Robinson 3 years ago that show that FlowSum can deal with rare subsets and demarkers pretty well. So for the pipeline, the platform is going to identify the clusters using FlowSum, even if the marker is dim. Dim is above 0. And then it will be able to use that for the cell subset assignment. And we have a few benchmarks of our own. 2 of them have been published. One in Frontiers of Immunology and the other in, I think it was Cell Reports, where we show that Astrolabe reproduces results from manual gating. So moving on to the next question, which is pretty related to Cytek. How does the Aurora perform with low intensity or dim markers like CD4 low on PDCs? Any advice on incorporating and incorporating them in panel design. And I'm guessing the question is incorporating low intensity markers in panel design.
So as for any flow cytometry panel design, we're going to treat those dim markers. As Janelle explained, we are assigning them to this tertiary group of antigens that we're paying extreme attention to. We're pairing them with bright fluors as we do in any flow cytometer, and we are taking very, very careful attention to the spread going into those fluorochromes to preserve the resolution of those antigens as much as possible. We start building our panel, fixing the assignment of fluors for those tertiary antigens. Those are the ones that we need to preserve the resolution as much as possible, and we build the panel to make sure around them, to make sure that we are not going to introduce any spread of any bright or high level expression of antigen into those dimly spread antigen. So I will say that from a panel design point of view, we're following exactly the same rules as we follow in the conventional cytometer. We take advantage of an instrument that has extremely good resolution in every single detector due to the nature of the detectors that are in the system and the really innovative optical design. So you are going to expect to see an improved resolution. And we see it especially in the far red. We have an extremely good resolution. So I will say that the combination of using this platform plus a good panel design should lead to extremely good resolution of the markers. In this data that was analyzed by Astrolabe, you will see that we got very good resolution of every antigen, including those that were dim, because of the platform and because of the panel design. So I think following the rules of panel design and having the right controls, as Janelle explained, is the key.
Yes, and I mentioned at the beginning of my half of the seminar that it's been quite a pleasure to work with Aurora data, and specifically, let's say, looking at marker like PD1, and sometimes you have PD1 bright cells, but more often than not, it's more of a gradient where finding the PD1 dim cells is important. I can safely say that with the Aurora, you consistently see that population, and it separates very well from the PD1 negative and the PD1 mid and the PD1 bright and so on. So next question is to Astrolabe. Can Astrolabe replace manual gating altogether? Oh, that's a pretty straightforward one. And yes, the goal of the platform is to replace manual gating and traditional gating. And the idea is that Astrolabe can take care of that for you. And as I mentioned before, we benchmarked Astrolabe against manual gating in numerous instances with different institutions, with different immunologists. And in all cases, we can say and we can show that Astrolabe reproduces manual gating very faithfully. So, so the goal is really to allow you as the biologist to focus on the biology instead of having to deal with traditional gating. And just to clarify, I have nothing against traditional gating, and I think that it works extremely well, especially if your panel is, let's say, smaller, between 6 and 10 colors. But once you go into high complexity cytometry, it is much more challenging to go through the complex hierarchy. And in these situations, platforms such as Astrolabe can standardize your workflow, reduce the amounts of errors in your experiment, and save you an inordinate amount of time. So I think we have time for one more question. One last question for Cytek. What are the biggest panels you have run for 3, 4, and 5 laser configurations?
So great questions. So for 3 laser configurations, the higher number of fluorochromes that we have effectively used together, and when I say effectively, is, as Janelle explained, without compromising the resolution. I think that has been something that we at Cytek have been very clear. We could add many colors in this beautiful instrument, but our goal is not only to allow for high parameter analysis, but high quality. And so we haven't pushed the number of colors to the max if we are going to sacrifice resolution. So with good resolution in the 3-laser system, we are up to 24. We feel very comfortable with that number. In a 5-laser configuration, we recently had a press release where we proudly announced a 40 color panel that we at Cytek optimize. That's the highest number we have done until now. The 4-color, I think the highest that I have run has been 36, I believe. So we are in that range of colors for all these configurations. Again, when we give a number of fluorochromes, people want to do more. And the only thing that we want to make sure is that you know that the instrument has the capability. You could use many, many colors, but let's always keep the quality of the data as our guiding mandate when we design the panels, when we QC our data. Maybe adding 1 color more can compromise the resolution of many of the markers, so it's not worth it. And that's why we feel so strongly about the principles that Janelle went through. So thank you so much for the questions and for your interest in our platform and in ways of improving the quality of the data.
Yes, so I think that's it for our webinar today. And I'd like to thank Guido, Maria, and Janelle from Cytek for joining us. And of course, everyone, all of our listeners for chiming in. So thank you all and have a wonderful day.